Hi sir, I am Rohan Itankar from Sonikar College of Pharmacy, third year. Our topic for elocution competition is Touch DNA, revolutionizing evidentiary DNA forensics. Touch DNA. Touch DNA is an advanced Touch DNA is an advanced technique in the modern world, widely used in modern justice systems in many developed countries. It aims to extract genetical characters from biological substances, especially from the cells that shed from the skin of the outermost layer of our hands when we touch a object or a surface. This method involves recovering traces of DNA which are usually found in very low quantity. Once the DNA is recovered, a person's DNA profile is created. Now, since dead cells are not really visible to the naked eye, the locating and recovering them quite be challenging. This method, which is used for the sampling procedure, collection, the preservation, removal of contaminants, quantification of DNA, amplification of genetical characters, and subsequent analysis and interpretation play a major role in showing how well the DNA works. Touch DNA analysis works. Touch DNA particularly refers to the bioavailability of the DNA or the presence of DNA through direct contact between an object or a surface. This helps the investigator of forensic sciences to determine who the perpetrator is. They collect the DNA left by the individual's handprints here and there. As we know, people people or many people shed their cells of outermost layer of our hands when they touch an object or some place. On the basis of the very famous Locard's principle, whenever a person touch or spush their hands on an object or a surface, they lose their cells from the lose their epithelial cells from the hands of their outermost skin. These cells are collected and their DNA is extracted from them, and the touch DNA is done, which can give the identity of the perpetrator. The importance of touch DNA is mainly found in crime scenes such as murder, rape, and there are many things which can be done and which can be convenient because of this touch DNA in forensics. Touch DNA is possible to be found in various areas including clothing, weapons, tools, door handles, and personal belongings. Touch DNA has gained increased interest in many infamous cases such as the acquittal of Timothy Masters and the John Bennett Ramsey homicide investigation. Though the lifting of touch DNA samples is not easy, there are many modes and methods such as the swabbing techniques, which is done by a wet cotton or a dry cotton. These are usually used for the smooth surfaces, but it's hard to collect DNA from rough or porous surfaces. The other method is like tape lifting, cutting and scraping, FTA card, etc. Interpretation of the results thus obtained from the DNA sample involves classifying profiles as full DNA profile, partial DNA profiles, mixed DNA profiles, or no results. Well, there are many sampling methods such as this next generation sequencing, or we can say quantification, capillary electrophoresis, etc. Several challenges can arise during DNA analysis, sampling, handling, and problems may occur due to various methods employed for the collection of touch DNA, such as swabbing, tape lifting, cutting, or scraping. Special care should be taken to minimize the contamination via personal challenges collecting the DNA and other sources at the crime scene. Contamination can also arise from improper handling. The primary goal of the forensic science is to enhance the assessment of evidence. Well, the success of touch DNA as a forensics tool depends on optimizing techniques from sample collection to profile generation, advances in methods, kits, instruments, and well equipped laboratories have improved the process, however,